have a minute or two more before our next talk. Sean Keenan will be up. Sean Keenan will be up next. But I wanted to remind people about the event we have tonight. Keith, you might have to remind me. It starts at 6. Is it 6 o'clock? Okay, yeah. So it starts at 6 o'clock. It's in the Marriott in the Waterside Grill. There's these little flyers on your chairs, and there's some in the back if you guys would like to attend. There'll be hors d'oeuvres and open bar and that sort of thing. It's a little group that sponsored us to get together and further the conversation on artificial reefs. So, anything else we need to add on that, Keith, or you're good? Oh.
Great.
All right, you ready, Sean? So up next, Sean Keenan is going to tell us about regional differences in reef fish assemblage structures between artificial and natural reefs in the eastern Gulf of Mexico.
Great. Thank you. Well, I'd like to acknowledge my co-authors. We all work at the Florida Fish and Wildlife Research Institute out of St. Petersburg, and this is demonstrating just some of the work that we do in a fishery independent survey in the eastern Gulf of Mexico. So it's become very familiar to the audience. The southeast U.S. reef fishes, they do support a diverse fishery. There are numerous species that are important for both recreational and commercial interests. And these fisheries come often heavily managed. Oftentimes the management that comes around those fisheries has a degree of political friction involved with that. So the longest term sets of data to feed into fisheries management generally come from fisheries dependent sources. And these sources do come with certain complications. They could be limited by changes in management due to size restrictions, bag limits. They could be influenced by changes in fishing effort, general fluctuations in fuel prices, or the relative change in the economy. And they could also be hindered by environmental causes, whether it be red tides or hurricanes that might influence the amount of effort. So due to this, there's been increased demand over recent decades for more fisheries independent data. These data are considered to be generally statistically strong. However, they do have certain downsides to them. They're generally costly. They oftentimes are done on large research vessels. They're also conducted by multiple entities. These can be both federal government agencies, state agencies, or private groups. And this complication brings in some differences in the data sets that are produced from these independent surveys. There could be variability in the gear type that's used, whether it be cameras or diver observations or capture gears. They will also be influenced by the particular statistical design or the sampling design that is used, and that the data that's produced can be different also. So FWC is just one of fishery independent surveys that are looking at reef fish in the eastern Gulf. The NOAA Pascagoula Lab has conducted a reef fish survey utilizing stationary underwater cameras. They actually do a Gulf-wide survey, although it's predominantly focused on shelf edge type features, areas that have large relief. They've been doing these surveys since the early 90s. In 2006, the NOAA lab in Panama City initiated a survey that's using the same type methodologies, and they were focusing on natural reef features both east and west of Cape San Blas. And in 2008, we got involved with addressing a spatial gap in the data working along the West Florida shelf. So we worked with the both NOAA groups to develop a sampling methodology. We developed the identical sampling gears in these stationary cameras. And all of these surveys are focusing primarily on natural reef habitats. In 2014, we got some funding from National Fish and Wildlife Foundation to expand our survey, both spatially as well as an addition of habitats, and that would be artificial habitats. So the Florida survey was expanded to go from the Keys all the way to Pensacola, and we extended our range of our survey to slightly deeper waters as well. And the grant that's supporting this effort goes through December of 2018. So the goal of these surveys is eventually to bring these data sets together. We're collecting the data in identical fashions. The goal is to come out with an integrated survey that can bring data sets that can be passed on to stock assessments. The strength we have is a common gear type. We're all using a stationary camera. This is just one of the gear types that's being used. We also use standardized hook gear surveys as well. There is a common challenge, though, and we've heard that oftentimes today, and that's about habitat. There was no comprehensive habitat map that existed in the eastern Gulf of Mexico when we started this survey. So we started by using a side scan sonar, and this provided us a cost-effective approach. It's a highly portable gear, as we commonly do our sampling on multiple vessel types, from large research boats to charter fishing boats, and it's a trainable technique. We have 15 to 20 staff that go offshore and collect the data in a standardized way, and we have those same staff that are in the office that are processing the data that come in. So to go over the spatial coverage of our survey, these look like little dots on the West Florida shelf in the eastern Gulf of Mexico. Each one of those dots is actually a side scan sonar survey that's designed at finding reef habitat. Our survey is conducted from 10 meters to 180 meters in water depth. And each one of our surveys is standardized in fashion. So this is a randomized design to pick the starting point of how the side scan survey functions. And all these surveys, the longer surveys shown here are the ones that are targeted at finding natural habitat. And the surveys are conducted perpendicular to shore. So in the Panhandle, they run north-south. In the Peninsula area, they run east-west. And then the surveys that are boxes here are actually surveys around artificial reef habitat. So this randomly selected reef is in the center of that, and the goal is to identify the reef habitat that's existing surrounding that artificial reef. So each of the surveys are approximately 2 square kilometers, and they cover 21 of these standardized grids that we use. And for each of the National Marine Fisheries statistical zones, we conduct about 100 surveys on targeted toward natural habitat, and 25 surveys targeted toward artificial habitat. So familiar side scan sonar waterfall. All the surveys are post-processed in the laboratory, and the final product is a 2 square kilometer geotiff that is manually delineated for habitats. So the polygons are drawn. You can see one habitats in green and blue here are manually drawn about the natural or artificial habitats. Now we've developed 36 habitat classes that anything that's identified is placed into one of these habitat features, and we follow the CMEX terminology as well as using some regional guidance as well. Not going to go into detail on each of these habitat types, but I'm focusing today predominantly on habitats from geologic origin, so low relief, natural hard bottom habitats, and habitats from an artificial origin. So for our camera, just our survey design, as I mentioned, we use stationary cameras. They're deployed on a specific location. They sink down to the bottom. They start recording. They record for half an hour underwater. And the data is read back in the laboratory for a standard 20 minutes. We also use the min count, or the max N, as Matt alluded to. So the maximum number of a species, identifiable species seen on the screen at one time becomes that number that's seen from that particular deployment. And that's a conservative estimate of relative abundance. These cameras are all stereo. They're stereo video cameras, so they allow for generation of length data without requiring the use of lasers. So the summary of the deployments that went into the analysis for today's talk, it's been limited to from the statistical zone 4 all the way to stat zone 10 and nearshore habitats. So everything from 10 meters to 37 meters, because that's predominantly where most of the artificial habitats are found in the Gulf. The data is from our 2014 all the way to 2016 surveys, and we had 681 sets on natural habitats, and that's shown in green, and 149 sets that were conducted on artificial habitats in red. And I should point out that the habitats are not stratified by habitat type before deployment. So anything that is identified as a natural feature is eligible to be sampled. Anything identified as an artificial feature is eligible to be sampled. These can be post-stratified later on as needed. So for the purpose of today's talk, we've pooled the data into regions. The Peninsula region, stat zones 4 and 5, the Big Bend here, as well as the Panhandle there. We conducted a series of multivariate analysis to look at the community data, and this is from the count data that was generated from the cameras. And this was done for 89 identifiable species. This is both managed groups as well as economically and ecologically important species. So a typical MDS plot, this is showing that we did see statistical difference in the species communities from each of those large regions and the two habitat types. So we see the geologic origin or any of the natural hard bottom habitats were grouped out here, Panhandle, Big Bend, and Peninsula. And then the anthropogenic or the man-made gear sets were shown here. So when we wanted to look at the species that were driving these differences, we used a similarity percentages analysis in Primer, and I'll be representing the differences by those particular regions. And abundances from natural habitats are shown in black, and the artificial reef data shown in gray. So for the peninsula, the differences were explained predominantly by increased abundances of gray snapper, tomtate, blue runner, and vermilion snapper. Moving north into the Big Bend, these differences were driven primarily by blue runner, gag, occurring greater numbers on artificials, as well as gray snapper and large numbers of tomtate. And then looking at data from the Panhandle, these differences were driven primarily by vermilion snapper, red snapper, and tomtate. Just want to point out a couple things from this area. Even though the lionfish numbers were greater in abundance from artificial habitats, the frequency of occurrence that we saw lionfish on the two different types of habitats were very similar. So, as a very common theme throughout the conference as we've seen so far, I'm going to take out a couple of these important fish species and look in a little bit greater detail. So from the Panhandle region, we wanted to look at a little bit more detail on gray triggerfish and red snapper data collected from this survey. So as I mentioned before, the stereo cameras allow for the generation of length data from software packages. And these data are summarized just for the Panhandle sets for those near shore areas of all the fish that were observed from natural habitats as well as artificial. And we did see significant differences in length frequencies of both red snapper, larger fish occurring on natural features, as well as gray triggerfish, larger fish occurring on the natural features as compared to the artificial. And these are all found within relatively similar depth ranges. The dashed lines shown here are just the estimated lengths at recreational harvest limits. So since this is kind of gearing toward an assessment type approach, we wanted to look at some of these preliminary data and just put out some very preliminary work on model index runs for red snapper and gray triggerfish. Again, this is just from the Panhandle habitats, just for these 3 years, pooling over all the habitat types. And generally, the patterns are very similar for both species, with the exception of red snapper in 2014, or higher from the artificial habitats. But you can see the patterns are pretty similar for both red snapper and gray triggerfish, even though abundances are greater. Well, these are corrected for means, excuse me. And we will have 2 additional years to tag onto this before the end of the project. So in conclusion, we are seeing that the differences in the communities are driven by increased abundances of species on the anthropogenic habitats as compared to natural habitat features. We are observing that red snapper and gray triggerfish, just the 2 that we looked at so far, are larger as found from natural habitats. And 3 years of data coming into an abundance indices do show similar patterns over time. So, also going to mention some future considerations. As we've heard, there are challenges for incorporating artificial reefs into a large-scale fishery-independent survey. One of these being the complexity and the diversity of the habitat types. Well, this is kind of jumping back to that graph or that plot, the table that showed all of our habitat types, and that's not really important except the fact that we have the habitats of geologic origin on this part of the chart, biogenic origin, which we didn't really get into here. Those are the reef building habitats, and then anthropogenic origin on the right side. When you look at it from a sheer area coverage scale, you can see that the geologic origin, and this bar is a flat hard bottom. So low relief hard bottom areas, just exposed rock. That's by far the largest coverage of habitat that we see, as opposed to the other features. However, when we're looking at it from a survey design approach, we categorize this by saying the number of sampling units, so the number of those individual grids in which a habitat occurred within. It gives us a little bit of a different story. We see habitats that are smaller in size. This orange bar represents potholes. Those are small habitats that are created or generated by grouper and tilefish species. They excavate portions of the hard bottom. And then you see this is unknown artificial reef. So if we identify something as man-made, but we don't know what it is, it's put into that category. And this kind of gives us an idea of how we can structure a sampling design to look at these habitats that may be more frequently found. So other challenges, we do know that have reports that artificial habitats may be ephemeral. There's commonly reports of artificial reefs may be in one area 1 year, and then after tropical systems, they may move or subside into the sediment. However, I've also had conversations with plenty of anglers and divers that will go off of Tampa Bay and dive on a 4-foot ledge 1 week, and then after a storm, they go back and it's not there. So it can be a similar type challenge on both aspects. There is an elevated exploitation on artificial reefs, and that's something that can be brought into our design. And then there's certain aspects of gear bias. Our stationary camera survey may not be the end-all, be-all approach. We're trying to get estimates of catchability, of what the cameras are actually seeing, what they're missing. We've seen plenty of video where red snapper are swimming higher up in the water column than our cameras are probably being able to see. So we're going to be addressing that by incorporating acoustics. We're going to, the plan is to deploy our cameras and run acoustic surveys around the area and look at relative changes in biomass distribution as compared to the cameras. Some fish are curious and attracted to these things underwater, and some fish are most likely wary of that. So this approach we're hoping would give us an idea of changes in distribution. So for acknowledgements, funding sources, as I mentioned before, there's a large number of staff members at the Institute that read hours and hours of side scan data, and as well as reading plenty of those hours of underwater camera data, and we couldn't show any of these data without their help. And then our colleagues at National Marine Fisheries, both in Panama City and Pascagoula labs. So with that, take any questions.
We have plenty of time for some questions.
Were you surprised that the catch rates for red snapper were basically the same on artificial and natural reefs?
The abundances were greater on artificial reefs. The max end counts were generally greater on artificial reefs over the course of the survey. However, they changed by region. We did see higher abundances of red snapper on natural reefs in the Big Bend. But if you're referring to the Panhandle areas, They were relatively similar in abundances overall with respect to habitat. A lot of the features in the Panhandle could be of the natural hard bottom is higher in relief, so it could be related to relief in that particular area.
You mentioned one of the aspects as a group as we're digging down to the natural bedrock and creating holes. One of the things that occurred to me is in the North Sea, at least, there are pipelines and communication wires on the bottom. And there is a tendency to, I think they're banned trawling in these areas. And also there's been gravel mining in some areas, and the trawlers avoid these areas because they're dangerous to their nets. So it occurs to me that could very well be creating holes in the bottom might be a cheaper way than creating reefs. If they're good for fish as a protection, or they need some kind of a structure to live within, they might find those areas very useful. Do you have any evidence of that kind of thing?
It's actually quite interesting is that not much is known about how these pothole features initiate. We don't know if it's a small amount of hard bottom that is exposed, and then once generally red grouper have been documented to do this. And they pick up sand in their mouths and they spit it out and they slowly excavate it. And then once that's done, or as it's occurring, a large suite of reef species are also finding that area and staying there. So it's hard to predict of where that's occurring. Generally, when we conduct one of our surveys and find these pothole habitats, they may occur in large swaths. They'll just be hundreds of dots within a side scan survey. We haven't done any large-scale trend analysis to see if that's occurring, but it's generally farther offshore. It's usually 70 meters or so of water. But it is definitely an interesting and a unique habitat type.
All right, thanks, Sean. We should probably move on to our next speaker. And I don't know. I don't know if we're going to give up on that. You might just give up on that this year if it doesn't.